day, everyone. I am Miss Rochelle Borja, your subject teacher in science. I am going to teach you General Biology 1 for this semester. Today, we are going to learn new things and it's gonna be fun. So, come and let's enjoy learning. Hello there! Today we are going to discuss lesson number 6, the cell cycle. For our objectives, first, characterize the phases of the cell cycle and their control points. Next, discuss crossing over and recombination in meiosis. Next, explain the significance or applications of mitosis and meiosis. Actively dividing eukaryote cells pass through a series of stages known collectively as the cell cycle. Two gap phases, G1 and G2, an S for synthesis phase in which the genetic material is duplicated, and an M phase in which mitosis partitions the genetic materials and the cell divides. Let us discuss the different stages of the cell cycle. G1 phase, metabolic changes prepare the cell for division. At a certain point, the restriction point, the cell is committed to division and moves into the S phase. S phase, DNA synthesis replicates the genetic material. Each chromosome now consists of two sister chromatids. G2 phase, metabolic changes assemble the cytoplasmic materials necessary for mitosis and cytokinesis. Next, M phase, a nuclear division which is mitosis, followed by a cell division which is cytokinesis. The period between mitotic division, that is G1, S, and G2, is known as interphase. Mitosis. Let us now discuss mitosis. Mitosis is a form of eukaryotic cell division that produces two daughter cells with the same genetic component as the parent cell. Chromosomes replicated during the S phase are divided in such a way as to ensure that each daughter cell receives a copy of every chromosome. In actively dividing animal cells, the whole process takes about 1 hour. Mitosis, although a continuous process, is conventionally divided into 4 stages: the prophase, metaphase, anaphase, and telophase. Prophase. Prophase occupies over half of mitosis. The nuclear membrane breaks down to form a number of small vesicles, and the nucleolus disintegrates. A structure known as the centrosome duplicates itself to form two daughter centrosomes that migrates to opposite ends of the cell. The centrosomes organize the production of microtubules that form the spindle fibers that constitute the mitotic spindle. The chromosomes condense into compact structure. Each replicated chromosomes can now be seen to consist of two identical chromatids or sister chromatids held together by a structure known as the centromere. Next, metaphase. The chromosomes align themselves along the metaphase plate of the spindle apparatus. When chromosomes become arranged so that their centromeres become aligned in one place, halfway between the spindle poles, the long axes of the chromosomes are 90 degrees to the spindle axis. The plane of the alignment is called the metaphase plate. Anaphase. Anaphase is the shortest stage of mitosis. The centromeres divide and the sister chromatids of each chromosomes are pulled apart or disjoined and move to the opposite ends of the cell, pulled by spindle fibers attached to the kinetochore regions. The separated sister chromatids are now referred to as daughter chromosomes. It is the alignment and separation in the metaphase and anaphase that is important in ensuring that each daughter cell receives a copy of every chromosomes. Telophase, the final stage of mitosis and a reversal of many of the processes observed during prophase. The nuclear membrane reforms around the chromosomes group at either pole of the cell. The chromosomes uncoil and become diffuse, and the spindle fibers disappear. The two sets of progeny chromosomes are assembled into two groups at opposite ends of the cell. Cytokinesis. Cytokinesis is the final cellular division to form two new cells. In plants, a cell plate forms along the line of the metaphase plate. In animals, there is a constriction of the cytoplasm. The cell then enters interphase, which is the interval between mitotic divisions. Let us proceed to meiosis. Meiosis is the form of eukaryotic cell division that produces haploid sex cells or gametes from diploid cells. The process takes the form of the one DNA replication followed by two successive nuclear and cellular division, which is the meiosis 1 and meiosis 2. As in mitosis, meiosis is preceded by a process of DNA replication that converts each chromosomes into two sister chromatids. Meiosis 1. Meiosis 1 separates the pairs of homologous chromosomes. During sexual reproduction, gametes combine in fertilization to reconstitute the diploid complements found in parental cells. The process involves two successive divisions of a diploid nucleus. In meiosis 1, a special cell division reduces the cell from diploid to haploid. Let's discuss the different stages of meiosis 1. Prophase 1. The homologous chromosomes pair and exchange DNA to form recombinant chromosomes. Prophase 1 is divided into 5 phases. We have leptotene, which is chromosomes start to condense. Next, zygotene. Homologous chromosomes become closely associated to form pairs of chromosomes consisting of 4 chromatids. Next, we have the pakitine, crossing over between pairs of homologous chromosomes to form chiasmata. And diplotin, wherein homologous chromosomes start to separate but remain attached by chiasmata. And finally, diakinesis, wherein homologous chromosomes continue to separate and chiasmata move to the ends of the chromosomes. Metaphase 1. The spindle apparatus is completely formed and the microtubules are attached to the centromere regions of the homologous. The synapse tetrads are found aligned at the metaphase plate instead of only replicated chromosomes. Anaphase 1. Chromosomes in each tetrad separate and migrate toward the opposite poles. The sister chromatids remain attached at their respective centromere regions. Next, telophase 1. The chromosomes now become diffuse and the nuclear membrane reforms. Each has a nucleus containing only one set of chromosomes or the haploid level in a replicated form. In most species, cytokinesis follows, producing two daughter cells. Cytokinesis, the final cellular division to form two new cells followed by meiosis 2. Meiosis 1 is a reduction division. The original diploid cell had two copies of each chromosome. The newly formed haploid cells have only one copy of each chromosomes. Let us proceed to meiosis 2. Meiosis 2 separates each chromosomes into two chromatids. The events in the second meiotic division are quite similar to mitotic division. The difference lies, however, in the number of chromosomes that each daughter cell receives. While the original chromosome number is maintained in mitosis, the number is reduced to half in meiosis. Prophase 2, the dyads contract. Metaphase 2, the centromeres are directed to the equatorial plate and then divide. Anaphase 2, the sister chromatids move away from each other and migrate to opposite poles of the spindle fibers. And telophase 2, the monads are at the poles, forming two groups of chromosomes. A nuclear membrane forms around each set of chromosomes and cytokinesis follows. The chromosomes uncoil and extend. And finally, the cytokinesis. The telophase stage of mitosis is accompanied by cytokinesis. The two nuclei are compartmentalized into separate daughter cells and complete the mitotic cell division process. In animal cells, cytokinesis occurs by the formation of a constriction in the middle of the cell until two daughter cells are formed. The constriction is often called cleavage or cell furrow. However, in most plant cells, this constriction is not evident. Instead, a new cell membrane and cell wall are assembled between the two nuclei to form a cell plate. Each side of cell plate is coated with a cell wall that eventually forms the two progeny cells.
Meiosis is a special type of cell division necessary for sexual reproduction. These are sperm-producing cells of a male crane fly, a species with only 8 chromosomes per cell. During meiosis, the cell divides twice, producing 4 haploid cells containing 1 copy of each chromosome. The first meiosis division separates pairs of homologous chromosomes, creating 2 haploid cells with only 1 set of chromosomes. The second division separates the chromatids in each chromosome. The end result is a production of 4 haploid cells, each with a single chromatid from each chromosome. When haploid sperm and egg unite during fertilization, the resulting cell is diploid, having received 1 chromosome of each pair from each parent.
And that ends our lesson number 6, the cell cycle. Thank you for watching.